Okay, this is Unit 6, Molecular Genetics. Remember that this is not a lesson, this is just a review. As per usual, there's a lot of stuff to try to condense within a 48-minute video. As I ask questions throughout this video, kind of like a Nick Jr. show, where the host asks a question and then there's this long, awkward silence, and he's like, oh, really? Great, let's go. I want you to try to answer those questions as I ask them throughout this video. Check those boxes in your mind. You either understand it or you don't. Hopefully you do. If you don't, we need to make sure you clear that up. As I record this, the AP exam is in 3 days. Let's begin. We start off with the central dogma of biology. A dogma is like a secular line of thought. And in biology, it basically means DNA creates mRNA, and mRNA produces protein. That's the central dogma of biology. And again, a dogma is like a one way of thinking. When it comes to the structure of DNA, remember that DNA is a nucleic acid. So I ask you guys here in class, if DNA is a nucleic acid, what are nucleic acids composed of? They're composed of nucleotides. What are the 3 components of a nucleotide? All right, hold on. So we're talking about DNA. I hear a nitrogenous base or just nitrogen base. What are the 4 nitrogenous bases for DNA? A, G, you said adenine, guanine, purine? Thymine and cytosine. Those are the 4, correct? We'll talk about purines and pyrimidines in a little bit. What else is there? There's a phosphate group. Remember, phosphate is PO4 with the -3 charge. Hopefully you learned that in chemistry. And what's the 3rd component? A pentose sugar. And what is the pentose sugar in DNA? Deoxyribose. On the first mock exam, the first question on the first FRQ said identify the components of an RNA nucleotide. You know, the first thing a student wrote, deoxyribose. Is that the sugar found in RNA? No, it is not. The D in DNA stands for deoxyribose. The R in RNA stands for ribose. All right, if I were to actually draw this, guys, I want to really show you how this looks, so please look up here. We have a phosphate group, then we have a pentose sugar, and then we have a nitrogenous base. So I'll put D for deoxyribose in there, and let's say that this will be adenine. Now I want to really show you how DNA is structured. What goes along the middle of DNA as if it were steps to a ladder? Is it the phosphate group, is it the pentose sugar, or is it the nitrogenous base? It's the nitrogenous base. So check this out. Here's another nucleotide. So let's say that's thymine. Now I have 2 nucleotides, okay? Here's another one. Now I have 3. The complement would be on the other side. Let me draw one more just for the heck of it. Here is a nucleotide that has cytosine. So let's make that cytosine. There we go. Okay. Now, the middle is the nucleotide. So between adenine and thymine, there's 2 nitrogenous bases. Or excuse me, hydrogen bonds, actually, that's what I meant to say. You can see how it's connected. If I really wanted to make this accurate, I would have drawn them upside down, but I'm not going to get into that much detail. So you know that we have phosphate right here. Then we have a nitrogenous base. There's adenine. So there's two, and I'll draw. I don't want to spend my whole video drawing this stuff, so let's see. I'll do one more. Okay, this portion here is the middle of DNA. Those are the nitrogenous bases. So what makes up the sides? What makes up the backbone? That's what we call the sides, the backbone of DNA. What makes those up? It's alternating sequences of phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, over and over and over. So as you can see, phosphate, sugar, phosphate, sugar, phosphate, sugar. Oops, wrong color. Sugar, phosphate, sugar. It's her. It just goes on and on and on like that. That's what makes up the side of DNA. We call that the backbone of DNA. All right. Let's go to the structure of RNA. RNA is also a nucleic acid. So what is the monomers? What are the monomers? No. You're telling me what the nitrogenous bases are when you say adenine and uracil. I'm asking what are the monomers of nucleic acid? Yeah, that's right. Monomers are nucleotides. So what are the parts of this was the first question on the first FRQ of the first mock exam. What are the what are the parts of a RNA nucleotide?
The nitrogenous base, which is AUGC.
Notice that U replaces T. That's how you can tell it's RNA. What else?
Another phosphate group. And then ribose is kind of sugar.
That is correct. Okay. Now, to save us some time, everybody, I am not going to draw an entire RNA molecule. It's generally the same structure, but RNA can be single-stranded or double-stranded. I'll get to that in a little bit. But no matter what it is, I want you to see that the backbone is made up of alternating sequences of phosphate, sugar. But this time, what is the sugar? Ribose. And remember that the bases are those of RNA. So you could have A, you could have U, you could have G, or you could have C. Okay? All right. A few differences between DNA and RNA, if I could include this. DNA is found in the nucleus only. Actually, let me rephrase that. Found in nucleus of eukaryotes. Where do you find DNA in a prokaryote? What's that area called? You notice I said area. The nucleoid. AP exam is this Friday. Where is RNA made in a eukaryote? Where does RNA come from? What is responsible for producing the RNA? No. RNA is made from DNA. And so in a eukaryote, if DNA makes RNA, where is RNA made? In the nucleus. Can it leave the nucleus? Yes. Where does it go? Where are the ribosomes found? Cytoplasm. Come on. Cytoplasm. They're made in the nucleus. They can enter the cytoplasm. The types of RNA that you had to know in this class was messenger RNA, transfer RNA, and ribosomal RNA. There is such thing, by the way, and I guess I'll throw this in here, microRNA, very, very, very small RNAs. All right. Structure, how it looks. In eukaryotes, like us, it's linear. DNA looks like this. And then in prokaryotes, do you recall how it looks? It's circular. Looks like this. Okay. Let's say about purines versus pyrimidines. A purine, here's how I remember it. Short name, large molecule. They look like this. It's like a hexagon and a pentagon. These are nitrogen ring molecules. This includes adenine and guanine. Adenine and guanine are purines. And then you have pyrimidines. Here's how I remember it. Large name, small molecule. This includes thymine if you're doing DNA, uracil if you're using RNA, and cytosine. Perhaps something you can use to help yourself remember this is that pyrimidine has the letter Y, and so does thymine, and so does cytosine. You're thinking, what about uracil? Well, you should know that uracil replaces thymine whenever you use RNA. All right, next is Chargaff's rule. This was a tricky one that was on the AP Bio mock exam that I provided you. This is only applicable towards double-stranded DNA or RNA. That's the key part. The percent of A must equal the percent of T or U if you're doing RNA, and the percent of G should equal the percent of C. So if you had RNA, this was the problem that was on your, I think on the second mock exam. It said, which of the following is representative of double-stranded RNA? Here's how it should look. If I have 40% adenine, guys, and I have double-stranded RNA, what percent uracil should I have? 40% uracil. What's 40 plus 40? 80. So what percent is left? 20. Okay, so I have G and C. What percent of G and C should there be? 10% each. This tells you that the RNA is double-stranded. Let me show you this one. How about this? Let's see, 45, 55, 65, 75, 80. 45, 55, 65, yeah, 80, so 20%. Is that double-stranded or single-stranded RNA? How do you know it's single? Does A equal U? No. There it is. You might say, well, G equals C. Yeah, I hear that, but it must be all or none. It's not equal. The amount of adenine does not equal the amount of uracil, so it is single-stranded. That means it doesn't have a complement on the other side. All right, let's get to DNA replication. When DNA makes DNA, this is known as semi-conservative replication. Semi-conservative replication is DNA making more DNA. Because this is DNA, guys, where is this taking place in a eukaryote? This is taking place in the nucleus. Let's make sure we have that understood. Okay, here are my parent strands of DNA. I'm going to just use 5 bases because we don't have all day. Okay. Here we have 5 prime, we have 3 prime, we have 3 prime, we have 5 prime. Do you remember which direction that DNA is read? Is it 5 to 3 or 3 to 5? It is read 3 to 5. So the bottom one will be read this direction, the top one will be read this direction. So what do we call this phenomenon where DNA is kind of like a highway where they're side by side but going in opposite directions? This is known as anti-parallelism, or you can just say that DNA is anti-parallel. That is correct. So the first thing that happens, what enzyme unzips the genes? Helicase does. So in step 1, helicase is an enzyme that actually opens up the DNA. It unzips it. So I'm just going to use my lasso tool here to open it up. Helicase is, what helicase does is it breaks hydrogen bonds connecting complementary bases. So now the bases have been separated. The next enzyme that you want to use is topoisomerase. What this does is it basically keeps DNA from winding up again. You don't want it to get all twisty while you're trying to copy. You want to keep it flat. That's what topoisomerase does. Also, another one is going to be single strand binding protein. This is not an enzyme. This is a protein. It prevents bases from rebonding. You don't want them to rebond while you're trying to copy them, so you need to keep them separated. That's what single strand binding protein will do. All right, now we have RNA primer. I'm going to use the color red here. An RNA primer is going to read the DNA 3 to 5. So what direction is the new half, the complementary strand, going to be created or synthesized in? Do you start on the 5 prime of the new strand or the 3 prime of the new strand? You're reading the template 3 to 5. So in what end do you make first on the complementary strand? You start on the 5 end. So here we're going to have a primer, and it's going to be the 5 prime. So if those are 2 G's, then this is going to be 2 C's. Over here we have a primer. If that's TA, then this is going to be AT. This is done by RNA primase. Remember I told you that when you want to paint a house or you want to paint your room, you need to start with a primer. A primer is like giving a fresh white coat so you can make whatever color walls you want. When there's a reason why coloring books are white and not black or blue or purple, because white generally serves as a primer. It's a place to start. When you want to make a puzzle, a lot of people like to put the edges together first. That's your primer. Prime means first. That's where you begin. So RNA primase sets down a RNA primer. And I made a little mistake I just realized. I want to clarify that mistake with you. This is RNA primase. So would that be a T? No, that would be a U. This is temporary. That U will get switched out. So let me make that abundantly clear. That uracil, which is technically RNA, is temporary. We're going to take it out. Okay? Now, do you remember what enzyme is going to start on those primers and lay down new bases based on what is already there? What enzyme lays down the complement based on the template? Anybody? DNA polymerase 3. DNA polymerase 3, and I really have to emphasize the DNA polymerase part. DNA polymerase reads DNA to make DNA. it synthesizes new DNA nucleotides, which are going to be complementary according to the template, which was already there. So the template says T, the complement says A. If the template says G, the complement says C. So here we go. My blue pen is going to be polymerase 3. Now we have two identical daughter strands. If you look at the two of them, top to bottom, they are identical. But you might be noticing, well, not for that primer. You're right. So there's another enzyme that's going to take away those primers and make it something permanent. This is going to be DNA polymerase 1. DNA polymerase 1 replaces RNA primers with DNA. And so here we go. Here comes RNA or DNA polymerase 1. It's going to erase this primer and make the proper DNA. It's going to come up here, erase this primer, and make the proper DNA. All right? And there you have it. You have two identical daughter strands based on what the original template was. This is how DNA replicates itself. It keeps half while making a new half. It keeps half by making a new half. Now, to get a little bit more extravagant here, let's see if you recall this. Let's say this is 3', this is 5', that's 5', that's 3'. You've opened up the DNA. The black strands are the template. Which strand is going to be going towards the replication fork? The bottom one or the top one? Look at the primes. Which way do we read DNA? 3 to 5 or 5 to 3? We're going to read the bottom one towards the fork. This is known as the leading strand. This is going to be assembled towards the replication fork. So what do you call the one that's built away from the fork? The lagging strand. Which one has the Okazaki fragments? The leading or the lagging? The lagging strand has Okazaki fragments. They're like little islands. Remember I told you Dr. Okazaki was a scientist from Japan. Japan's an island. They're little islands of DNA. And then what enzyme seals those gaps on the lagging strand? Ligase does. Ligase seals any, that was the last question on the multiple choice portion of the second mock exam. You had DNA that looked like this, guys. Check it out. You were asked what enzyme could fix this. The answer is ligase, because what part is damaged? Is it the middle or is it the sides? No, it isn't, Kevin. It is not the middle. Or Jason. Look, you see this right here? See those gaps?
I was talking about like the middle.
Those are the side, the middle. When you refer to the middle of the DNA, you're referring to the bases, like the parts of the ladder that you grip, that you walk on. That's what the middle is. The sides are the backbone. And what is the sides made up of? Alternating sequences of what?
Phosphate groups.
And what enzyme can seal those phosphates and sugars? Ligase does. That's right. Ligase seals those gaps. Very good. Okay, let's move on to transcription now. Transcription is going from DNA to mRNA. So where does this take place? Guys, when you see something with DNA, you need to automatically know it's taking place in the nucleus if it's a eukaryote. If it's a prokaryote, do they have a nucleus? No. So it's going to be taking place in the nucleoid, which is just a region. Does transcription take place in the nucleus? Why? Because it involves DNA. That needs to come naturally. It shouldn't be a roadblock. It shouldn't be a mental gap, a writer's block, or a wall. if it involves DNA and you're talking about a eukaryote, it must take place in the nucleus. Period. There's no exceptions there. All right. So here we have DNA strand. I'm only going to make this 4 bases long because we don't have time for all this stuff to go. This is going to be like a month and a half in one video. What enzyme is going to open this up? Helicase is correct. So let's do that. Let's open it up. All right. What enzyme creates RNA based on the DNA? Do you know that one? What enzyme will read the DNA template and produce an RNA complement? Which one? RNA polymerase. That is correct. Now, there's 2 strands here, everybody, and there's only one you're going to copy. So here's what I'm going to tell you. This strand on the top, we're going to call the non-template strand. So is RNA polymerase going to use this one? No. That one's not going to be a factor. This one down here, we're going to call our template strand. This is the one that we are going to make complements for. And so the next enzyme is RNA polymerase. This synthesizes RNA nucleotides according to the DNA template. Now, one thing I didn't mention here, 3, 5, 3, 5 prime. Okay, which direction are we going to read the template strand? From left to right or right to left? How do you know? Because the 3 is on the left. That's right. So here we go, guys. Here comes RNA polymerase. It sees an A. What's it going to make? U. Then what? A. Keep going. G. And then what? C. That's right. And so here's your 5 prime. Here's your 3 prime. Ladies and gentlemen, this is your RNA. There it is. That is going to be able to leave the nucleus. Any questions so far? All right. So let's pretend that this RNA is over here now. This is my pre-mRNA. When I say pre-mRNA, it's not ready yet. Not ready yet. Here's my 5 prime. Here's my 3 prime. Do you remember what parts of pre-mRNA that you cut out? You cut out the introns. Very good. So you have to remove the introns to turn it from pre-mRNA to a final mRNA. So that intron is gone. That intron is gone. So here's, if we go down, how do we get these 3 exons together? What's that word? Rhymes with slice? You splice them. That's right. So let's do that. Let's put them together. Okay, now it's still not done. We have to add a couple more things here. On the 5 prime end, we're going to have a guanine cap. We call it a 5 prime cap. It's like a bunch of guanines. And on the 3 prime, do you remember what we put? We put a bunch of adenines. This is known as the poly A tail. So this is a 5 prime cap. Over here is a poly A tail. The 5 prime cap and the poly A tail help the mRNA survive in the cytoplasm. Eventually, RNA can be degraded in the cytoplasm. These 2 components help to protect it. This is now known as a final mRNA transcript. This all takes place in the nucleus, but it is now ready to leave the nucleus. It's like a little bird. It's finally grown up and ready to leave the nest. It can then go out of the nucleus to the cytoplasm. What is it going to go to in the cytoplasm? Jason mentioned this earlier. Where does it go in the cytoplasm? Where? It goes to a ribosome. What is made in a ribosome? Proteins. Good. That gets us to translation. Now, I'm going to really abbreviate this one, but this is where mRNA synthesizes protein. This takes place on a ribosome. What type of RNA is a ribosome made up of? Ribosomal RNA. Very good. What do you call those 3 base codes that are written on the mRNA? They're called codons. What is the start codon? AUG. So for instance, every RNA, mRNA, begins with the start codon AUG. This codes for the amino acid methionine. Do you remember how many different amino acids there are? There's 20. Now I'm going to pull up, give me one moment. I'm going to pull up a codon chart that we can refer to here. Just give me a second to find a good one online. If you just go to Google and type in codon chart, you can find millions of them. I've found one. Just waiting for it to load up. And it's taking too long, so let's pick a different one. That's taking too long too. So here's what we'll have to do. Just use the main screen on Google here. All right, so let's go back to your notes. Let's say that we have an mRNA that reads like this, guys. I'm just going to include, I'm going to make up some codons, okay? It says AUG, CCC, UAU, UGA, okay? These are codons. What is the opposite of a codon? What carries the anticodons? tRNA. So the tRNA would have UAC, GGG, AUA, ACU. Now, these anticodons are going to be carrying the amino acids, but I want to make something very clear to all of you. I remember when I was in school, I made this mistake. Whenever you're trying to figure out the amino acid sequence, you need to refer to the codons, not the anticodons. So let's look for AUG. I already know what it is, but just for the sake of showing you, if you look at this screen, you look on the far left, you have A, then U at the top, then G over here on the right. That's MET. I know that that stands for methionine. They just abbreviated it. So this is methionine, AUG. Let's look up CCC, see what that is. C, that's proline. See, it says PRO. That's going to be the amino acid proline. And then we have UAU. Let's look at that. UAU, that's tyrosine, TYR, short for tyrosine. And then we have UGA. UGA, oh, that's a stop codon. So now how many amino acids long is this protein? It's 3. Stop is not an amino acid. It just means that you're done. What would happen if G right here, actually, let's go back up. That this C, what was that? Did I have a C there? Okay, let's say that was placed with an A. And then that means that's going to be a U. What type of mutation is that known as? It's a substitution mutation. Let's find out if it's actually going to change something. So we instead of having CCC, I have CAC. Let's see if that changes it. CAC, yes, it did. It changed it to histidine. Should histidine be there? No. So this is going to be a missense mutation, where it actually does affect the proper sequence of amino acids. This is where it actually does have an effect. A silent mutation would be if there is no change, and then a nonsense mutation is if you prematurely end the protein. Let's say it was supposed to be 50 amino acids long and a mutation causes it to be 15. That would be a nonsense where it prematurely ends. If my last name was subject to a nonsense mutation, it looked like this. That's nonsense. It was ended prematurely. All right. You had your hand up, Lucas?
Yeah, I was actually going to ask if it would be nonsense.
A nonsense is where it ends prematurely. A missense is where you have a substitution. It changes the flow of the amino acids. And then a silent is where there was mutation, but it did not change anything. You guys okay so far? I know that this is a lot of stuff. This is more than a month's worth of material wrapped up into just one short video. Tell me again, what type of RNA, how about this? Here's a good question. When making a protein, tRNA, rRNA, and mRNA all provide different parts. Which type of RNA provides the proper code? mRNA. Which type of RNA provides the facility to make the protein? No. rRNA, the ribosome. Do you remember how many parts of a ribosome there are? 2. You're thinking of the 3 chambers. The A, the P, and the E chamber. There's 2, a large subunit, a small subunit. The rRNA provides the facility. What provides the actual amino acids? The tRNA. So mRNA provides the code. tRNA provides the amino acids. And rRNA provides the facility to do it. So the way I talked about it in class is that mRNA is the cookbook, tRNA is the ingredients, and rRNA is the kitchen, it's the facility. All right, let's go to gene expression. Remember that most genes are turned off. They're methylated. Signals can activate them, or we actually say express them. We would call this acetylated. If you said demethylated, that would also be fine. So a few days ago, I was asking a student, and I'll ask Jason right here. Jason, in the cells in my brain, are those somatic cells or gametes? Somatic. They're somatic cells. So do my brain cells have a full copy of my DNA or half? A full copy. Jason, do I have the genetic information to build a kneecap present in the DNA in the cells in my brain?
Yes.
Yes, I do. But are those genes expressed or not being expressed right now?
No.
They're not being expressed. If we know how to do this, we could turn on the genes in the cells in my brain to build a kneecap, and you could build a kneecap in my head. Now, we don't want that, but that's actually, we do have that understanding of DNA. Most of our DNA is turned off, and it requires special signals at special times to turn it on. When you guys are like 3 years old, 4 years old, you had the cells for armpit hair. They're in your armpits. But we all know that 3 and 4 year olds don't have armpit hair. But then there's a gene that turns on that then makes that armpit hair. That's part of puberty. The DNA was there the whole time. It was just off. And then at a certain stage of puberty, it turns on. It received a special signal. The DNA was there all along. It was there all along. So gene expression is just about activating genes or shutting genes down. We call this inducible DNA. Inducible DNA means that it is expressible. It can be activated. We have a student I mentioned this before in my first period, she broke her arm. Normally our bones are not repairing themselves because they're fine. But in the case that she has broken bones, her genes to fix the bones were activated. And as soon as her bone is healed, those genes will turn off. You no longer need them. Sometimes when we have cancer, genes are turned on when they shouldn't be, and the signals that are turned on, the ones for growth. When we talked about prokaryotes, we talked about the operon model. The operon model is where a prokaryote can actually regulate its own DNA. We have the regulator gene. Then we have the promoter, the operator, and the structural genes. The structural genes are the genes that you actually want to transcribe. Okay, check this out. Here is RNA polymerase. If I have RNA polymerase here, it's going to get onto the promoter, and then it's going to move down the DNA. Do you guys see an inhibitor anywhere to stop this RNA polymerase? No, so this means that the operon is turned on. But let's put my RNA polymerase back here, and I'm going to plug in a inhibitor, big I for inhibitor. When this RNA polymerase gets onto the promoter, and then it tries to go down the DNA, can it get past that inhibitor? No, it cannot. So this means that the operon is turned off. Remember that inhibitors can shut things down. Promoters want to make things happen. Now, what we did in class is we did two operons. We did a tryptophan operon, and we did the lactose operon. Tryptophan is an amino acid. Whenever you have tryptophan, it needs to be turned on because whenever you have tryptophan, it's turned off. This makes tryptophan. Do you need to make tryptophan if you already have tryptophan? No. So when tryptophan is present, the operon is off. And when it is absent, the operon is on. Think of it like this. When you're going back to watch this video, hopefully you'll watch this again. The tryptophan operon makes tryptophan whenever you don't have tryptophan. It's like making yourself a sandwich because you don't have a sandwich. If there is tryptophan, you don't need to make more tryptophan. So when it's on, when it's present, it's turned off. And when tryptophan is nowhere to be found, the trip operon will be turned on. Now, the lactose operon produces lactase. Just by looking at the name lactase, what does lactase break down? It breaks down lactose. Under what conditions do you think the lactose operon is going to be on? When there is or is not, when lactose is or is not present? When it is present, because you need to break it down.
When it's off.
It's on when lactose is present. Because if there's lactose there, you need to digest it. You need to metabolize it. It's turned off when lactose is absent. There's no need to break down lactose if there is no lactose to break down. So don't forget that whenever it's off, the inhibitor is plugged into the operator. Whenever it's turned on, the inhibitor is removed from the operator. Yes?
There are other enzymes these two, right?
Yeah, but these are the two we had to know for class. All right, we're only a few minutes left. We have about 6 or 7 minutes. Let's talk about mutations. Mutations can cause new phenotypes. Blue eyes, blonde hair, pale skin. Those are all mutations in humans. All humans derive from Africa. Africans have darker skin, darker features. And then over the course of thousands and thousands of years, there were some mutations that happened. And now we have all different races and cultures and appearances. That's mutation. As I mentioned earlier, there's a few types here. We have silent, which means there's no change. There's no effect. There is a change, but there's no effect. I'll use my name for an example. If you had a silent mutation in my name, you would spell it C-U-R-T. It still says Kurt. No change. A missense is where it does have an effect. So if you change my name to Bert, that would definitely be a missense mutation. A nonsense mutation is where it prematurely ends. So let's say you did K-U-R. It didn't Kurt. It didn't finish. It had a premature stop codon. Mutations can be beneficial. They can be neutral. And they can be detrimental. I have a mutation where I was born without wisdom teeth. That's great. I like it. I also don't have a molar on the bottom right of my jaw. I was just born without it. It's in my family. That's kind of neutral. It doesn't affect how I eat. I eat just fine. But I also don't have to have 4 wisdom teeth pulled from my face, so I kind of enjoy that. But some people can have a detrimental mutation, like maybe an allergy or maybe the inability to smell. It's just weird things like that. Remember that mutations are the primary source of genetic variation. Now, we obviously have crossing over and things like that, but this is how we get a lot of differences. And nature, remember this part, you'll learn this tomorrow with evolution. Yes, evolution is contingent on mutation, but the environment the environment determines whether those mutations are going to be beneficial or not. A bear that develops a mutation for white fur in the jungle would not be great. But a bear that develops a mutation for white fur in the poles would be great. So the environment determines whether the mutation is an advantage or not. Guys, remember with viruses, you start with viral DNA, then you go to viral RNA, and then you go to viral proteins. This all takes place in the nucleus of a host. Retroviruses, what do you start off with? Viral mRNA, and then you go to viral DNA. What process is this called? RNA to DNA. Reverse transcription. And then you go from viral DNA to RNA. What is this called? No. DNA to RNA is just transcription. And then RNA to viral proteins. Okay, we're running low on time, so the last little part I'm going to have to ask you to, I guess, really just summarize. Let's see, we have biotech. The ones we did in class are gel electrophoresis. There are very brief videos online. You can look these up. They're actually very simple, guys. You just need to refamiliarize yourselves with them. We have polymerase chain reaction. This is known as PCR. We have transformation. You did this example with the bacteria. Remember, you were cutting things out and you were adding a human gene into a bacterial plasmid. That's called transformation. And then DNA sequencing, where you actually read the DNA and you translate what the codes mean. This was done in the 90s. It was called the Human Genome Project, where businesses and governments from around the world work together to decipher the entire human DNA code. Everything, figure out what everything is. These are the biotechs I would recommend that you look up. Again, they're very simple, and you guys have that foundational knowledge. You've been taught this already. You just probably need a little refresher. So I would go on YouTube or watch a quick video on just what these are to refamiliarize yourselves with them, because there were problems for all of these on the mock exams that you received in April. Okay, we will stop there.